Good morning. Today I will be talking about On the Threshold of Injustice, Manipulating DNA Evidence. Legal aid defender Clinton Hughes just described the New York versus Nick Hillary case and how keeping out the DNA evidence contributed to the defendant's acquittal. In my portion, I will discuss how software was used to interpret the DNA mixture data. What is a DNA mixture? The left of the slide shows multiple people. The middle shows their DNA combining to form a mixed DNA sample. The right shows how DNA science unmixes a mixture. A crime lab first extracts DNA, then amplifies it using polymerase chain reaction, or PCR. The lab then separates amplified DNA by fragment size. The amount of each fragment is measured by fluorescence. Short tandem repeat, or STR, data has a peak for each allele fragment. A computer can separate STR data into the genetic types, called genotypes, of the people who contributed their DNA to the mixture. What is probabilistic genotyping? In old-style DNA identification, the data was so definitive that it gave only one genotype. We still see this with reference samples from cheek swabs or blood draws. But most DNA evidence is now a mixture of two or more people, and the DNA data are less definite. Many different genotypes can explain the data, so there is uncertainty in the genotype. Scientists use probability to represent uncertainty. Probabilistic genotyping means assigning probabilities to possible genotypes. Bayesian probability uses computers instead of people to interpret data. The Bayes approach updates probability through a mathematical examination of evidence. The human alternative can introduce bias and error. Bayesian genotyping handles noise by statistically modeling the baseline. With lots of data, Bayes can use all the peaks. With low-level data, it considers all the genotypes, often finding exclusionary match statistics. For distorting factors, like PCR stutter, Bayesian genotyping reaches into the data and mines the evidence to resolve issues. Data variation is addressed by using more variables. The goal is accurate and objective computer comparison. However, some genotyping software implements less mathematics. Removing sections of math from a Bayesian system diminishes its capability. When the baseline isn't modeled, people instead apply arbitrary thresholds. When not using all the peaks, analysts discard data. When considering only some genotypes on low-level data, human operators set dropout levels that reduce exclusionary power. For stutter and other artifacts, limited software can't mine evidence data. Instead, calibration is done on old data unrelated to the DNA in a case. Inadequate modeling of variation can make software just give up and give no answer. An incomplete model requires help from a human operator, a person who makes subjective choices. Subjectivity introduces human choices and the consequences of those choices. While objective probability can be fair, gambling against stacked house odds is not as fair. Human decisions have bias that undermines objectivity. That unneeded subjectivity can lead to an unfair scientific outcome. In the Hillary case, DNA items were recovered after the death of a 12-year-old boy. Of the 150 analyzed evidence items, the police were particularly interested in the boy's fingernails. The prosecution's genotyping software gave a match statistic of 10 million, connecting the fingernails to the defendant. The matching component was an 0.4% or 1 in 250 minor contributor to the fingernail mixture. Rule 702 governs the reliability of scientific evidence in federal courts and in most states. The rule requires sufficient data, a reliable method, and the reliable application of that method to the data. Challenges to admissibility can keep potentially unreliable scientific evidence out of the courtroom. In this case, the general reliability of the mixture software was acknowledged. However, the defense mounted an as-applied challenge to the method's reliability on the particular fingernail DNA evidence. The sufficiency of the fingernail data was questioned. The DNA mixture ratio was 1 part in 250, less than 1 human cell. The peak heights of the minor component were in the range of 30 to 70 relative fluorescent units, abbreviated RFU. These heights are low on a detection scale of 0 to 10,000 RFU. The reliability of the software's mixture interpretation was considered in different circumstances, as applied to different types of DNA data. The developer had previously conducted a validation study. In that study, the smallest mixture ratio tested was 10 times higher, 1 in 25, than the minuscule 1 in 250 ratio seen in this case. The study's DNA samples contained many human cells. The software used a peak height threshold of 30 RFU. The right side of the slide shows validation information provided by the developer for the admissibility hearing. On top of their monochrome data figure, I have superimposed some color markup. Along the x-axis, I show the number of cells that correspond to DNA picogram quantities. On the right, at the level of 3 cells, the system seems to work fairly well. When the minor component comes from a donor, labeled in green, the circles above the x-axis show positive match statistics in the quadrillions, and on the bottom right, below the x-axis line, non-donors produce exclusionary statistics under 1 in a quadrillion. At this level, the software can separate donors and non-donors by their match statistics. Moving leftward on the x-axis, there are fewer than 2 cells. We start seeing some non-donor X data points rising up to the x-axis, showing an uninformative match statistic of 1. And on the left, near the y-axis where the number of cells is between 0 and 1, there is commingling of donor and non-donor. The match statistic of a donor, shown as an O, becomes similar to that of a non-donor, shown as an X. When there is less than 1 cell, the software's match statistic cannot separate donors to the DNA mixture from non-donors. Let's zoom in near the y-axis, where there is less than a cell's worth of DNA. The study examined DNA levels that were actually used in this case, where the mixture ratio is 1 in 250. I've outlined that data in a red rectangle. Again, the X's indicate non-donors whose DNA is not in the mixture, and the O's show true donors. The donor match statistics, inside the red rectangle, cluster around a log LR match level of 2 zeros, or an LR of 100. Note that the reported statistic in this case of 7 zeros, or 10 million, is far away from these validation study match values of 100. On the far right of the figure, the x-axis shows a DNA amount of one cell. So on the left, by the y-axis, there is less than half a cell's worth of DNA. At this extremely low quantity of DNA, the developer's validation study highlights the software's limitations. Based on the calculated match statistic, it cannot distinguish donors from non-donors. At a 1 in 250 mixture ratio, the software's likelihood ratio doesn't tell us whether or not somebody's DNA was actually present in the biological evidence. A threshold of 30 RFU was used in the validation study, but a higher 50 RFU threshold was applied to the case data. A lower threshold includes more data, which is potentially exculpatory. This is because considering more possible genotypes can lower the match statistic. Reducing the statistic can convert a large inclusionary number to a small exclusionary number. A non-Bayesian statistical model is incomplete, and so choices must be made and entered by a person into a computer program. When using an incomplete computer system, applying a peak height threshold introduces subjective choice. What threshold should the software operator choose? Does the threshold choice make any material difference to the match statistic outcome? We answer that here. The table shows likelihood ratio results at different RFU threshold levels, listed in the first column. The defense team explored these alternative thresholds, applying the software to the fingernail mixture data. The prosecution expert's threshold choice was 50 RFU. The second column shows the likelihood ratio outcome when making additional stutter peak data choices. The third column shows the LR outcome when not making those data choices. The reported prosecution result at 50 RFU, when making stutter data choices, was 15.5 million. When the defense tested lower thresholds of 30 or 40 RFU using the same software parameters, that inclusionary number dropped to 0, indicating exclusion. On a peak height scale that goes up to 10,000, 10 RFU is 1 part in 1,000. That sliver of threshold change made all the difference between inclusion and exclusion. There were other parameter assumptions and user choices, but close threshold values of 30 and 50 RFU led to diametrically opposite conclusions. Different choices gave different answers. The software did not agree with itself. Here is the STR data from 3 PCR amplifications done at locus D8. The x-axis shows allele size, while the y-axis shows peak height. The 3 amplifications are colored in different shades of blue. What allele data was chosen as input for the software? How do those subjective choices affect the match statistic? The software operator chose to input the victim's alleles 10 and 12, shown as brown V's in the bottom chart. In the next blue row, an F letter shows that foreign allele 16 was also chosen. The D's in the green row show that the defendant's alleles 12 and 16 were also selected for program input. But, marked with red X's, the last row shows 5 potentially exculpatory alleles, 6, 9, 11, 13, and 14, that were not entered by the prosecution expert into the software. Omitting that allele data can artificially inflate the match statistic. More allele data opens more allele pair possibilities, with the potential to spread probability across more genotypes. This spreading can lower the match statistic, changing inclusion to exclusion. With incomplete software that isn't fully Bayesian, choices are required, and these subjective choices can radically alter the outcome. The defendant's genotype was unrelated to the allele data used at locus D18. The entered data alleles were 9, 10, 11, and 12. There is no 17 allele in these data, but the defendant's genotype is 1717 at this locus. His alleles are not in the data. The data at this locus support exclusion, not inclusion. But at D18, the software gave an inclusionary match statistic by assuming that the defendant's two 17 alleles had doubly dropped out of the data. The software could include him. It gave 15% weight to the Q dropout alleles and divided that by 14% for random Q alleles. 15 over 14 gives a likelihood ratio bigger than 1, an inclusionary number for exclusionary data. Turning up a dropout parameter can increase match statistics for alleles that aren't there. Users choose their dropout parameter to account for missing data. Larger dropout parameters can convert exclusionary data into an inclusionary statistic. The judge's ruling noted the absence of internal validation, which was needed to guide user choices. The ruling said that no internal validation studies were performed. As a result, wrote the judge, the expert was forced to pick and choose data and input parameters. Therefore, the judge precluded the prosecution software from evidence. Fully Bayesian modeling eliminates user choices, but incomplete probability software forces a user to choose their data and parameters. The judge decided that those choices should be guided by validation studies. Such information can help human analysts reduce subjective bias when using limited software. Relevance Rule 403 is a related admissibility consideration. An interpretation method may be reliable on sufficient data, but unreliably applied to insufficient DNA data. A judge can balance the probative value of an unreliably applied method against the danger of unfair DNA prejudice that can mislead a jury. The software's legal relevance was not directly addressed in this case. Here are some recommendations for trial attorneys, whether prosecution or defense, on discovery requests for forensics interpretation software. Know the range of software applicability. Discovery should request validation studies to see how software users set their parameters. Request the user, procedure, and training manuals. These show how the technology is actually applied in casework. Papers, reports, and mathematical descriptions can be helpful, particularly if you retain an expert. Understand the data choices people make. How much subjectivity or bias intrudes when human analysts run incomplete, non-Bayesian software? In a case, request all DNA data. This is available as electronic computer files. Request a copy of the working software. Your expert can then apply the method to the DNA evidence in your case, varying thresholds, data choices, and parameters. You want to know the range of possible answers, not just what the lab reported. If you don't have their software, get a continuance until it arrives. As seen in this case, a small change in threshold can radically alter the conclusion, changing inclusion to exclusion. Run the software on the case data to verify the lab's results. Run other genotyping software on the case data to see the match statistics of other methods. There are a dozen genotyping programs available. There is much online information about genotyping on our website and YouTube. We have posted publications, lectures, CLE courses, webinars on how objective genotyping works, admissibility rulings, case outcomes, related news, and more. Thank you very much.